Thank you for watching this presentation. We have nothing to disclose. Central acetylcholine is involved in inhibition of the micturition reflex through brain muscarinic receptors. Acetylcholine also stimulates nicotinic receptors, which are reported to inhibit the micturition reflex in the brain. However, the brain mechanisms for nicotinic receptor-mediated inhibition of the micturition reflex remain unclear. In this study, therefore, we investigated brain mechanisms for brain nicotinic receptor-mediated inhibition of the rat micturition reflex, focusing on brain nicotinic receptor subtypes and brain GABA receptors. This slide shows experimental procedures. In urethane anesthetized male Western rats, a nicotinic receptor agonist, epibatidine, was intracerebroventrically administered 30 minutes after central pretreatment with each nicotinic and GABA receptor antagonist. Continuous CMZ was started 60 minutes before the administration of each antagonist. Then I'll show you our results. First, we investigated the effects of centrally administered epibatidine on urodynamic parameters, intercontraction intervals ICI, and maximal voiding pressure MVP. As you can see, this agonist dose-dependently prolonged ICI without changing MVP. Next, we investigated the effects of central pretreatment with MEC, a nicotinic receptor antagonist, on the epibatidine-induced response. Pretreatment with MEC dose-dependently suppressed the epibatidine-induced ICI prolongation. These results indicate that stimulation of brain nicotinic receptors induced ICI prolongation. In the brain, alpha-7 and alpha-4 beta-2 are representative subtypes of nicotinic receptors. Therefore, we investigated the effects of each receptor antagonist, MLA, and alpha-7 receptor antagonist, and DH-beta-E and alpha-4 beta-2 receptor antagonist on the epibatidine-induced ICI prolongation. Central pretreatment with MLA dose-dependently suppressed the epibatidine-induced ICI prolongation, while DH-beta-E showed no significant effect. These results indicate that centrally administered epibatidine induced ICI prolongation through brain alpha-7 nicotinic receptors. We further investigated the effects of centrally administered PHA and alpha-7 nicotinic receptor agonist on ICI and MVP. Similar to epibatidine, this agonist PHA dose-dependently prolonged ICI without changing MVP. These results also support that stimulation of brain alpha-7 nicotinic receptors induced ICI prolongation. As we know, GABA is a representative inhibitory neurotransmitter. Therefore, we investigated the effects of SR, GABAA receptor antagonist, and SCH, a GABAB receptor antagonist, on the epibatidine-induced ICI prolongation. Central pretreatment with SR or SCH significantly suppressed the epibatidine-induced ICI prolongation, respectively. These results indicate that centrally administered epibatidine induced ICI prolongation through brain GABAA and GABAB receptors. This slide shows summary of our results. Centrally administered epibatidine and nicotinic receptor agonist prolonged intercontraction intervals ICI without changing maximal avoiding pressure MVP. The epibatidine-induced ICI prolongation was suppressed by central pretreatment with MEC, a nicotinic receptor antagonist, and MLA, an alpha-7 nicotinic receptor antagonist, but not with DHbeta-E, an alpha-4 beta-2 nicotinic receptor antagonist. Similar to epibatidine, centrally administered PHA, an alpha-7 nicotinic receptor agonist, prolonged ICI without changing MVP. The epibatidine-induced ICI prolongation was suppressed by central pretreatment with SR, a GABA-A receptor antagonist, and SCH, a GABA-B receptor antagonist. These results indicate that brain GABA-A and GABA-B receptors are involved in brain alpha-7 nicotinic receptor-mediated inhibition of the rat micturition reflex. That's all. Thank you for your kind attention.